Well, hi, my name's Oliver Saunders Wilder. I'm a postdoctoral fellow at the Simon Center for the Social Brain at MIT, and I'm excited to get to be here with all of you today. And I was going to talk a little bit about effective computing in the classroom based on my experience, which has been applying effective computing technologies over the last 10 years to understand, mostly in context with children, what we can learn, especially in the field, by leveraging these types of technologies. So first I wanted to talk just briefly about a little bit of guiding theory, because obviously anytime you reduce something to measurement, you're always making some choices about how you're going to conceptualize it, and I like to be upfront about my choices in terms of understanding what I'm going to show you after this. So this may well be familiar to many of you, but I like to think about affect in terms of the circumplex model, which is something first, it's been around since the 1980s, and essentially it's a means of sort of conceptualizing affect as this two-dimensional plane in which you have arousal as one dimension, which we can think about somewhat as the intensity of an experienced emotion, and then valence, which goes from negative to positive. And for a given emotion word or category that we might talk about, like anger, you can place this on the plane. So anger is typically thought about as being a negative emotion that's high in arousal. Fear is another one that would also be negative and high in arousal. Excited would be high in arousal but positive in valence, for example. So it's a relatively simplistic thing, but it's important as we think about sort of bringing computational techniques to bear on this to think about how we're modeling it. And related to that, my work is very much informed by some of the work in neuroscience that's been taking place recently, in which affect can be sort of understood as the conscious perception of one's own physiological condition. So that is to say that the experience of affect is something that's being constructed in the brain based on the brain's information and predictions about your body's physiological state. And that actually is helpful because it gives us some insight into how we can measure it. And so without going into a lot of detail about this, the sort of theoretical perspective that informs a lot of my work here is this idea that, as has been pointed out by Lisa Feldman-Barrett, who's a former colleague of mine at Northeastern, there's this sort of theory that emotions and what we think about as emotions are constructed. And her argument would be that the emotional experience that we subjectively have is a construction based on this sort of affective substrate, so this sort of general sense of positive or negative and what our physiological arousal is, bringing that together with sort of context and the act of categorizing that internal experience, and that produces sort of an emotion. And I think the easiest way to think about this is that it's analogous, in a sense, to the way in which visual predictions or visual information gives rise to the sort of subjective experience of vision. So we all we have this experience that we're seeing, me or whatever we're looking at, or this beautiful room, and that's all very seamless from our sort of perception, but what's actually going on in the brain is that at a series of different hierarchical levels, predictions are being made about what types of sensory input are expected, and then the error between those predictions and the sensory input. And that experience of trying to categorize and sort of make sense of the sensory input is what gives rise to our ability to say we see a particular thing or a particular color, or that's a table. And in a similar way with affect, there's evidence to sort of suggest that essentially what's going on is that we're categorizing these internal signals together with what we know about the world and the context and the things we're experiencing, and that gives rise to our sort of subjective emotional experience.
What are interoceptive predictions?
Sorry, so there's a lot on the slide. So interoception is, exteroception is the sort of act of in the brain of perceiving sensory information from the outside world. So things like the light that's hitting our eyes and the experience of touch or sound or those things. Interoception is the equivalent thing, but for sensing the internal state of the body. So for the most part, sort of your sense of what your heart rate is. And there are reasons relating to the way in which these cortical pathways are constructed that you'll notice we have a sort of, people talk about a sort of gut sense. You have this sort of less precise sense of the state of your internal milieu that's sort of a little bit less nuanced and specific than the equivalent exteroceptive senses. I won't go into detail about that, but essentially interoception here is talking about perceiving your internal state. And so, in that sense, you perceive some physiological changes, your heart rate, goes up, you start to sweat, and this perception of these physiological changes is related to your experience of feeling afraid, for example, or feeling tense or stressed out. Yeah, no problem. So why does this matter? This matters because as we try to think about measuring affect, and then a little bit later in this presentation, measuring affect in a social way, we need to understand, because affect is inherently a subjective construct, and because we know that there are limitations to asking people about their own affect, because people, especially kids, have varying degrees to which they can effectively self-report that. And you can't effectively ask somebody to self-report continuously. Actually, there have been attempts to do that using dials, and it turns out having that creates a secondary process that can be distracting, and so it can actually interfere, and you can get a lot of measurement reactivity. So the reason this matters is because we're trying to think about what we want to measure to understand this sort of experience that the person is generating inside themselves, and to get some insight into that. So I want to talk briefly about some of the technologies that I have experience with for measuring both sort of dimensions of that two-dimensional plane I was talking about. First, I want to talk a little bit about arousal. So arousal is a tricky thing to measure non-invasively, but there's been a lot of progress in that in recent years. And one of the, I think, best ways that are out there right now to measure this is using peripheral autonomic physiology. And what that means is measuring things like electrodermal activity. So electrodermal activity, we all know that when we're nervous, our palms get sweaty. And that happens because you have a higher distribution of eccrine sweat glands, which evolutionarily probably were designed to help you grip things. And when your cortical arousal goes up and your body essentially prepares you for, we often talk about this in terms of fight or flight, but this is happening all the time to sort of maintain your homeostasis and match your body's readiness state to the demands of the environment, your eccrine sweat glands dilate. And so that experience that we know of our palms getting sweaty, you can actually measure that electrically in very, very fine degrees. So you can measure things that are very much imperceptible in terms of your sweat level on your skin, but you can see sensitive electronic changes from this that can tell you a lot about how someone's sympathetic nervous system is ramping up or down. And in addition to that, there are the measures that people may be familiar with for measuring cardiac function, so measuring changes in your heart rate or your heart rate variability. Heart rate itself is not an autonomic indicator, but you can derive things like heart rate variability that are. And there's two main technologies in ambulatory measurement that you see for this. One is electrocardiography, or ECG. The other is photoplasmography. ECG is electrical, so we have the electrodes on your chest. Photoplasmography uses reflected infrared light. So essentially it's doing a fancy version of shining a flashlight through your hand and noting that the color is red. And if you could look very carefully at the changes in that color, you could actually perceive when your heart is beating.
Except for that last one, is PVG. Is that like a lie detector?
Yes, so all of these things are commonly used in lie detectors, and I will say, for my part, the more that I have learned over the years and more experience I have gained with physiology, the more skeptical I am of using a lie detector for anything.
I thought they were proven to not predict anything.
I am not a fan of lie detectors because at the end of the day, all of these things tell you something about arousal. The relationship between arousal and lying is a very complex thing that depends a lot on the person, the situation, and all of these things depend a lot on interpreting the outputs of this. So I think they're very informative signals, but I wouldn't use them for deception detection personally, though I don't work on that. Important to note here is that generally looking at these things, you can't distinguish a positive versus a negative affective state, which is to say someone's arousal level when they're excited versus when they're stressed out or frightened may look similar. But you can tell a lot about, in very sort of fine degrees, how much they're reacting to a specific situation. And then with all of these things, additional information is really needed to assess the adaptiveness of a given arousal level for a given context. So that is to say, measuring that someone has high arousal, however we're going to define that in whatever units, typically isn't that useful unless you know something about the demands of the context. There are lots of contexts in which high arousal is adaptive, and there's also lots of contexts in which it would not be, and vice versa. Some examples of some of this wearable sensing. Wearing one of them right now. Sometimes in talks I put it up on the screen so you can see how nervous I am or not. I won't do that today, but you know, we are measuring this in case we want to review it later. So I would say there's sort of two classes of these things. One are these wrist-worn devices, like the Empatica device. These have gotten so much better over the years. When I first started doing this 10 years ago, literally there was not any device on the market that did this, which is why we invented one and commercialized it, because nobody else had. Now, wonderfully, there are a number of these devices, and increasingly things like this are built into your watch, and though not all of them have the research quality data we'd like. But this is becoming increasingly available. The E4, which I'm wearing here, measures both photoplasmography and electrodermal activity, as well as 3-axis accelerometry, so you can get some idea, especially with kids, about what they're doing based on the motion patterns that it's capturing as well, which can be another interesting area. And then there's cardiovascular sensors like the ActiWave Cardio. This is a small wearable thing about this big that's worn on the chest. I have used both of these things successfully with young kids and young kids with developmental disabilities, so this is absolutely feasible, and I will show you some examples of this from a typical kindergarten class that I collected data on last year.
And these are recording your activity continuously, or?
They're recording continuously. The E4, you can easily record a full day, I think probably 2 full days, without having to recharge it. The ActiWave Cardio is a little bit less. I think you can only record up to about 4 hours on a single charge on those. But yeah, with the wrist-worn devices these days, you can get very much continuous recording over a long period of time. So you get a lot of data, which is great. This is an example of data that I recorded in a kindergarten classroom in Massachusetts last year, part of a pilot. I'm not going to go into a lot of detail about these data specifically. It's just to show you, for anyone who is familiar with physiology data, you actually can get relatively good quality electrodermal activity data on young kids who are moving around and doing a variety of different tasks. These are not kids who were told to sit still and do this. These were put on them at the beginning of the day, and they were told to go about their kindergarten activities. So we did ask them to avoid the water table because the sensitive electronic equipment does not love that. And what you can see here is that you get a fair amount of variety, both in terms of the types of patterns that you see across these different children over time. This is about a 2-hour period here, hour and a half. And you can also see that you get differences in level and also differences in variability across kids and across time. And all of these things are useful in understanding what's going on internally for a child if you brought this together with the context, and we usually would. Something I want to mention that's on the horizon as something that I think is an interesting and exciting technology that is probably applicable to some of these sort of simulation types of things that are being done now, and I think will soon be applicable to an even wider variety, is non-contact physiological sensing. These are both technologies that were developed by colleagues of mine at MIT. The one on the left actually uses your cell phone, and it can work to some degree even when it's in your pocket, and it uses the accelerometer, which measures very fine changes in how it's moving, which was put in there originally so that when you flip the screen, everything turns. Well, my colleague Javier figured out that you could actually extract things like respiration and heart rate from that signal that's being collected automatically, because those motions are being carried in the motion of your body. So if you carry your cell phone in your breast pocket, in many cases you could actually get a lot of physiological data from that without needing any specialized equipment whatsoever, which is pretty cool. Another technology originally developed by the two folks on the right, it's Ming Po and their colleague, and this is called CardioCam. This is also out there commercially now. And it uses a webcam, a typical webcam like the one that's built into your computer, to sense heart rate by essentially exploiting what I was talking about with the flashlight through the hand. It's looking at the light that's reflected off your face, and there's subtle and imperceptible changes in how that light is being reflected and the color of that light that change as blood pumps through your body. And so you actually can pick up heart rate as well as respiration and sometimes pulse ox, so the oxygen content of the blood, using these technologies.
What's the degree of accuracy compared to like standard?
It's a great question. So it depends a lot, and I could point you, if you remind me afterwards, I could point you to papers on both of these that actually have stats on it. I don't want to pull them off the top of my head. This one is, I know, both I would say are pretty accurate under fairly controlled conditions. So I know that if you have good lighting and you're sitting in front of a screen and you're not moving a ton, that the CardioCam is pretty accurate, pretty highly correlated. I think, I forget what the mean error is, but it's pretty low. It's in the, under 10 beats per minute, I think. And then I would say it's similar, I believe, for this, but again, I think it depends on things like where you're holding the phone and where it is relative to your body. But both have been characterized. So I just want to briefly touch on a couple of important considerations and limitations when using this type of technology. The first thing that we've found really consistently is it's important to treat each participant as their own control, because the dynamic range of physiological responses varies across people and time for biological reasons that don't necessarily pertain to the psychological questions of interest. So things like how well hydrated you are that day can affect the absolute level of your EDA. So when we look at the kids that I showed you earlier, what we're interested in is not so much that this kid's at 25 and this kid's at 29. It's that they're varying, the way in which they're varying over time and the way in which they're varying relative to their own baseline. So we typically try to collect longitudinal measurement as much as possible. So if we can get multiple hours for the same person, that's great. If you can get multiple days, that's even better. This lets us characterize sort of what this person's dynamic range is across time. And then we also tend to try to select a baseline comparison point. So if there's something where all the children are doing something similar that's relatively low energy or that's relaxing, then that provides a comparison point across children that we can compare to some other point of interest during the piece here. And then the other piece is that ambulatory data like this is inherently noisy, and so we've had to develop and approach this with slightly different techniques than people in traditional psychophysiology would, who oftentimes would focus on a single, 10-second response to flashing up an image on screen, and if the participant moved at all, they were removed from the data set. We embrace the fact that this data is going to be somewhat noisy, and we try to plan the ways in which we analyze it and the ways in which we sort of quantify it with an understanding of that and choosing statistics that are robust to that. So now I wanted to touch briefly on valence, and then we'll get a little bit to social interaction. So measuring valence, I would say, is in some ways less straightforward because it's harder to directly measure what's going on in terms of how someone is conceptualizing their physiological state relative to positive or negative. Computer vision analyses of facial expressions and nonverbal behavior are currently one of the more common means of assessing this. So this is using things like things that will automatically try to label the facial muscle movements on your face, as well as things that look at stuff like posture, approach, withdraw, how if you're interacting with someone, for example, whether you're moving towards or away from them, and those kinds of things. And advances have made it possible to automate a lot of this. Not 100% perfect, but there's, again, compared to when I started doing this stuff 10 years ago, an amazing amount of technology out there, especially even in the commercial space, to try to automate some of these pieces.
Can you measure like two-person interactions? Right now you're dealing with a single person.
Yes.
But now you're talking about valence and arousal, for example, for one person, but now they're interacting.
Yes, you absolutely can. And I will, in fact, I will give you some examples both for this, and then I'm going to talk a lot about how we do that on the physiology side as well. But I think that is one of the places where this is really interesting. So, before I leave this slide, I just want to mention that one of the pieces that people who do work in the space don't always talk about is that mapping from a facial expression, which is a particular visual configuration of the face, to how it means you feel is not straightforward. So for example, smile, which is one of the things that we all think about, there's research that my advisor and colleagues at the Media Lab have done that shows there are actually smiles for every emotion under the sun. There's angry smiles, stressed out smiles, unhappy smiles. There's lots of different ways that we use smile. And so just knowing that it's a smile doesn't mean you can say this person's happy, or they're having fun, or they're enjoying something.
Just as a clarification, I mean, you mentioned before webcam-based techniques to determine arousal, but how different is that from what you're saying here? I mean, what is the interaction between the two?
So you could use, in this case, you could use the same camera. It's just that you would be applying different types of technology to extract information from it. So, and the information that it tells you is different. So typically, there's a little bit you can tell from facial expressions about sort of this arousal or intensity dimension, but typically people in the space don't think of it as being a particularly good measure of intensity or arousal. It's much easier to distinguish based on facial expressions, whether you're a human or a computer, positive versus negative, than it would be to say based on the facial expressions how positive or how negative. So I think actually one of the places where there's lots of opportunity is combining these technologies, and as much as possible we always try to do that. It's not always applicable in every setting, but I think when you can, you're better off. So it's just important to keep in mind when you look at these technologies generally that the fact that you can recognize facial expressions doesn't necessarily mean that you can, you're making an inference in going from the recognized facial expression to making some sort of an affective claim. And the last here is that these are significantly influenced by the social context, which I'm going to talk more about in just a minute. So just briefly, this is one of these areas where for strange reasons, industries unrelated to this research have been a huge boon, namely the Microsoft Kinect and the GoPro and proliferation of those, because they actually turn out to be really nice little cameras in both cases. And so you can actually leverage things like the Kinect to do 3D imaging, where you not only are getting the image that you can see, but also depth. This lets you get really important things like measuring, if you were talking about two different people, how close they are to each other. It also lets you automatically extract some of these nonverbal information about posture and things like that. And so it's getting increasingly straightforward and affordable to collect these kind of data, which is really exciting. So I'm just going to show this really briefly. So this is an example, not using a Kinect, but actually just using these head-worn glasses that, again, were actually developed, I think, for skiers who wanted to take action video as they skied down slopes. But they turn out to be really interesting for collecting interpersonal interaction data. And so what's going on here is essentially I've got an adult wearing those glasses, and then I've run a computer vision algorithm on the output of that video, and I'm identifying times when that child is essentially making eye contact or is looking and orienting towards the adult's face. So times when they have sort of shared social gaze. And this is something that when we talk to practitioners and to people who study this is something that is of real interest when you're looking at these video, but instead of having an army of grad students who have to code it, you could theoretically use this type of approach to automatically extract that type of information in certain settings.
You
So
talked earlier about how it's like people mask their emotions, their behavior. Are kids maybe less good at filtering that? Like, could you kind of code their facial to know their valence easier?
That's a good question. So I don't know to what degree that has been specifically studied, but I'll tell you based on my own experience working with young kids, kids for the most part pick up social norms really quickly, much more quickly than I think most of us give them credit for. And so I think that you will start to see kids who are taking into account sort of display rules about how they're displaying different types of affect, probably earlier than a lot of us, I would guess, would think. Probably less than adults, but I think that I think it's always important to think about the context in any case, because there may be, as I'll get to in a minute, I actually think there are other social reasons beyond sort of inhibiting that the social context has a really strong influence on actually people's experience of affect and emotion. So this is just one example of the kinds of stuff you can do. This is another example done by a colleague of mine, Natasha Jacques, and others in the lab that I'm in. This was looking between adults at who were having a conversation, so they were randomly paired. They had this conversation. They used the Kinects in a room to capture both people's facial expressions as well as both people's sort of body posture and where they were in the room. And then they ran deep learning algorithms on it to try to predict bonding, which was measured using a sort of standardized test, I think originally for therapist-client types of interactions. And essentially, there are just two interesting things that they found in addition to the fact that they could... It's a self-reported measure from both people about their experience during the conversation. So these data are extracted from the video, and then the machine learning was attempted to use these sort of extracted computer vision metrics to predict the self-report data on people's self-reported experience during the conversation, how connected they felt with the person they were having a conversation with.
So essentially the facial expression software is trained to self-report previous self-reported data?
Yes. Essentially, in this case, what they were doing is extracting the facial expressions with software that just extracts facial expressions, and then doing a different layer where they took the facial expression information about the people's body posture and where they were in the room, and then gave that to a machine learning model that tried to say, okay, based on the facial expressions, based on this data, can you predict who's going to have experienced more bonding and who's going to have experienced less bonding on this self-report measure. And so just two interesting things that they found. So one is that they found that for the participant who experienced the lowest bonding, they found this mismatch, so that when the person expressed sadness and anger in terms of their facial expressions, the partner was responding with smiles and smirks and sneers. So these sort of, there was this mismatch between the expressions that they found correlated really highly with, at least in this case, what was going on there. And I think that speaks to, for me, the transactional nature of what's going on here. And then they also found that the distance between participants' bodies was strongly correlated in conversations with high bonding. So people were maintaining this sort of interpersonal distance and being responsive to one another in the conversations where they reported higher bonding, whereas they weren't doing that to the same degree in the lower bonding conversations. So this is just meant to be really an example of the types of information that we're collecting and the types of things that you could use it to explore. So then just briefly, some considerations.
So if you had something like the slide, the video you put up, you have two agents who are quite asymmetric. I mean, there's the mother and then there's the child. How would you measure the kind of bonding going on, some kind of relationship there? In this case, yeah, you don't have a validity check for the child. I guess you could for the parent, I'm not sure.
Yeah, so I think that's one, I mean, that's always one of the challenges when working with young kids, and in some of my work working with kids who actually can't communicate necessarily. I think that the short answer to that is that I tend to like physiological means of looking at this question because of the fact that they get around this issue of sort of the social display. So for example, with the little kid, there's also, you could ask questions about what's his motor development like, and is it harder for him to hold his head up and orient? And I've seen this when I used to work with kids clinically. So there are I think there are a lot of layers to understanding what the nonverbal behavior is telling you. And that's definitely trickier when you have really asymmetric relationships with this. And this is one of the places where some of the physiological measures that I'll talk about in a moment, I think could have a real role to play.
Can you also take audio measures?
Yes.
So somehow, so you do, so simultaneously.
Yes, so we always collect audio. I do not personally do a lot of affective research with audio, but I know people who do. And you certainly can extract meaningful information there. And again, I would say what you find is that I think the research is biased towards older kids and adults. And so as you go to younger and younger children, it gets more challenging to figure out what variations in tone of voice mean, and to find software that's been trained and validated on that population. But there's definitely...
No, but in your video, you could at least determine what the mother was doing.
Oh, no, absolutely. We always collect audio. I don't have it in this particular video just because I use it primarily just as an example of that computer vision piece. But yes, so all of the things, including all the physiology studies that I'm showing you, we're also collecting video and audio, sometimes from as many as 5 different cameras.
And how does the physiological measurement, say, again, going back to younger children, how would it align with something like when a woman was breastfeeding her child, or some kind of action of cradling the child or something? We know sort of independently we think that's a good thing maybe for the kid, but would you, if you had these measurements, how would it show up here, for example?
So I have not done that study, though there are, we did at one point apply for a grant to do a study on that. I think that everything that I have learned would suggest that I think that things like that are actually really critical. The sort of physical proximity and comforting that's going on between parents and a young child are actually probably very critical to the child's development of their own sense of affect and of that child's affect regulation, because I actually think that there's a great deal of co-regulation that's going on in those settings. And I actually think there's physiological reasons why, for example, hugging is a thing that we primarily do between people that we have some degree of intimacy with, right? And it's an expression of that. And if you think about what's going on there, when you're being held that way as a baby, when you're hugging someone, you're actually doing things that allow you to perceive their changes in their physiology in a way that you can't as readily when you're at a distance from them. And I actually think that my reading of the neuroscience literature suggests that we are very much wired to sync up, and things like that actually facilitate that synchrony. So I strongly suspect that if you were to look at that in mothers and children breastfeeding, that you'd find that there's a very high degree of physiological synchrony that's taking place there, and that in fact there's a great deal of that that's happening early in development, and that it's very critical.
Later, there's a guy at UCLA named Daniel J. Siegel, he's a neuroscientist, and his field is interpersonal neurobiology. And so he's looking, basically says we all have a neural Wi-Fi. We're sort of pretty much picking up on things in the same way. I don't know how much he's measuring it with this, but there's a lot of more therapeutic type of things.
I know of his work, but I haven't actually met him. But I'll have to take another look at it, because I think that there are a number of people who are starting to see these things, and I think that it's one of the things that I'm really excited about. So just to close up sort of on computer vision here, I think the things you want to keep in mind are that it's difficult to judge intensity from facial expressions and nonverbals alone. The data tend to be discontinuous because in any sort of a real-world environment, you don't have a perfect view of people all the time, and that's just sort of a reality. And so when you plan your study and plan the way that you're going to leverage this, we always do it sort of assuming that. And then, again, these sort of nonverbal and facial expression mappings to an affective or emotional state are complicated and may vary across individuals and across cultures. And then, unlike physiology, facial affect and nonverbals can be consciously sort of suppressed to some degree. And so that's something to keep in mind. So coming to the sort of social piece that we've been dancing around here. So I think that although a majority of extant research on affect generally focuses on individuals in isolation, and that tends to be in many areas how affect is talked about, we consistently find that social and interpersonal context has very significant influence on both measured and self-reported affect across a variety of settings. I will never forget doing focus groups years ago when I was working in industry and finding that the part where everybody introduced themselves and said a little bit about themselves was by far sort of the most interesting responses that you were seeing physiologically. But we've seen this across a wide variety of settings. And so in order to understand both intra and interpersonal affective dynamics, I think it's really key to capture the interpersonal context and to analyze it in conjunction with individual level data. And I think that's true even if what you're interested in is the individual level data, because I think the social dynamics have a big role to play. And so essentially you end up with two choices in doing this. Either you can try to control the social context, which is what we do with things like simulations or where you have them interacting with an agent, which tells you a certain set of things, or you can measure it. And in the work that I'm going to show you that we've done, we pretty much always choose B and try to essentially measure it at as much scale as we possibly can so that we can understand it and look at it afterwards in interpreting the results of our studies. I would make the argument, as I sort of alluded to, that affect is inherently social. And so there's a growing body of research that actually suggests that the very act of predicting another person's internal affective state has the effect of activating the same neural systems that would be active if one was experiencing that affective state themselves. That means when I'm watching somebody experience something, my brain and this is true also if I'm watching somebody throw a baseball, the same pieces of my brain that would light up if I was throwing a baseball are lighting up. Now obviously it doesn't take it all the way through to the fact that I start moving, but you will find that as I start nodding, people start nodding. And this is and somebody coughs in a theater and everybody coughs, right? And so I would argue, I think that we're increasingly finding that this is not a unique thing about the affective system or about the motor system, but in fact is actually sort of a rule in terms of how the brain is organized and a natural consequence of the brain's organization. And so when we are watching other people and when we're present with other people, we are automatically and continuously simulating their affect in addition to ours to some degree. And I think this is one way of explaining the affective contagion work that we are all, that you see all the time in everyday life, in which, watching someone feel upset has an effect on us. Watching someone else have something exciting or positive happen to them makes us feel good, even though nothing has changed in us. And sometimes it's not even a person we know. It's an imaginary person in a movie. And yet this still has a profound affective impact on us. And I think this is really interesting. And I, about 8 years ago, set out to try to figure out a way to measure it that would be scalable and that we could use in the settings that were of interest to me, like in classrooms with children and in everyday interactions. And so measuring interpersonal affective dynamics with physiology is, I think, a really interesting approach because, one, it gets at the heart of what we think is driving conscious affective experience, and because two, you can do it relatively unobtrusively, and you can do it continuously across a really wide variety of settings using ambulatory psychophysiology equipment. So interpersonal physiology, which is sort of a burgeoning field, is the study of relationships between people's physiological dynamics. Although it's relatively young, the existing findings sort of across the board indicate that matched physiological activity occurs between two or more people across a range of contexts. And for the remainder of this, I'm going to mostly refer to this as physiological synchrony, though it's important to note that that doesn't necessarily mean that two people are moving together. It can also mean that two people are moving in a way that's responsive. So it really is talking more about sort of the mutual influence here. And then this has been found to correlate with a range of psychosocial constructs, and this is everything from mothers and children and mothers and infants to research between therapists and clients and ratings of how moment-to-moment empathic the client and the therapist, rated that moment, to sort of marketing and a variety of other fields. And so given these findings, interpersonal physiological interactions are theorized to be sort of ubiquitous social processes that underlie some of this observable behavior, and in fact, I would argue the conscious experience of affect that we have. Now, since I don't have a ton of time, I'm not going to go into a lot of detail here, but I'm happy to answer questions about this. But I'm going to talk a little bit about a model that I've developed that builds on work done by Ferrer and Helm and some others that essentially leverages dynamical systems modeling to try and quantify this when we're looking at multiple physiological streams. So you think back to the data I showed you with the teacher and the students, and you imagine taking a student and the teacher, and we're trying to say how much is each person's physiology influencing the other person. How much can we predict about one person's physiology just from knowing the other person's physiology in this setting? So we use a regression-based technique with a dynamical systems model and a windowed approach that creates a time series that captures this interdependence over time. And then I use a hierarchical regression to essentially extract a metric that is the interpersonal variance explained. And the reason I'm mentioning this is because the study examples I'm going to show you in just a minute are all going to use this metric as sort of the effect size. So in order to isolate this, we're comparing essentially two different models, one of which includes the other person, one of which only includes sort of some intrapersonal variables. And so the value we get out represents the percentage of variance in one person's physiology that's explainable or predictable by changes in their partner's physiology. And something to note is that this turns out to be directional. So you actually can find that you can have settings in which one person's physiological changes predict their partner, but the opposite is not true to the same degree.
So you're fitting a time series model.
Yes.
Like an autoregressive model for each child.
Yes.
For each party, sorry.
Yes.
And then you're fitting cross lags across is that?
Essentially. So we fit a system of equations. So they're fit together because the errors may be correlated. But essentially, yes, we fit a model based on essentially the person's tendency to return to their own personal baseline and the difference between them and their partner. And then we fit two sets of models for each person. And then we fit the same models, but without their partner's variable. So just looking at the variable that sort of talks about their tendency to return to baseline and their randomness. And then we take the difference in effect size in variance explained from those models. And that is the metric that you get here, which essentially says what percentage of variance in my signal was predictable by changes in your signal.
But there must be some elasticity, a lot of flexibility in choosing exactly how many lags to include in these models and what the cross.
Yes, so you'll find actually a surprising number of these things do the best at zero lag. And I think that actually is because I think that a lot of what we're doing is predicting other people's affective response, not necessarily
So it's
perceiving
simultaneous.
it.
I mean, it's like
Yes.
contemporaneous. You're just putting each in the other. I see.
Yes, we've done both, and there are some circumstances, and when one lag turns out to be better than another lag is something that I'm really interested in, but still need to do more research on to be able to say confidently what it tells us. My suspicion is that people who have a relationship and know each other are better at doing this faster because they are better at predicting the affective response. So I think about this with my wife. I'm much better at predicting how she's going to react to something than I would be with somebody who was a stranger who I was interacting with for the first time. And I think I probably start to have a physiological response to something that happens before she may have that response or right as she has that response because I'm predicting how she's going to respond to something. So I think that may be one of the reasons this works in zero.
But here you have continuous time data or
Yes.
discrete time data?
So the input, the input is ultimately discrete because you're sampling EDA some number of times per second. Usually we're sampling a lot faster than we really need to be to capture the changes. Most of the things I'm going to show you, I think, are at 8 samples a second. We often downsample a little bit, particularly for these interpersonal analyses, because we're usually trying to look at slightly longer term trends on the tens.
But then when you talk about lags, I mean, how many lags do you talk about? And how many seconds or how many minutes?
Seconds. Seconds is the most that I've ever done. I think 10 seconds is probably the most that I've looked at. I haven't studied the lag issue exhaustively. simply because this is actually a relatively new technique that's still, we're in the process of submitting a paper on this, and it was developed as part of my dissertation work. So I was, up until last year, as I was finishing my dissertation, I was focused first on putting it into practice on some of the data that I've collected that I'm going to show you. So I actually am really interested to go back and do a more fine-grained exploration of what the lags are, how much they may change across time for people, and what that may be telling you also about how an interaction is developing. So I think it's a rich area for future exploration.
But do we know that it says the interaction between these two, or what? Maybe they're just confronting a common stimulus.
Yes, that certainly
So that-
could be the case.
Yeah. So that's why I'm wondering if you can somehow standardize the environments so that that's even there. The standardization wouldn't solve the problem, I guess, but at least it would get rid of you could say at least at this benchmark environment. I'm getting this kind of covariability.
I would agree. So I'll both show you an example of where we attempted to do something like that in just a second. And also, I'll also say, though, I think that, so one, I would say I think of this not as a sort of technique that in and of itself, achieving a particular value can ever be your end goal. I think of this as not unlike the correlation coefficient, which is a useful measure, but it tells you nothing meaningful unless you have a good study design.
But given those previous graphs that you were showing us, I mean, the thing that immediately came to mind was like some form of spectral analysis. So you could have like a long-run trend, short-term. So you have a lot. You really can sort of decompose this into different frequencies. And I just wonder, you could imagine covariance, the cross spectrum at some frequencies would be very different from the spectrum at, say, long-run frequencies and so forth. So that's why I think it would be interesting maybe to use some of these those are non-parametric techniques.
Definitely.
Yeah.
And that there are some people who have done that more often with heart rate data than with electrodermal activity. Though I think a technique like that I think has been applied to electrodermal activity. I think that the trade-off with any of these things becomes a question of window size. So one of the things I like about this technique is that it allows you to specify different window sizes. And so we can look at it in a 10-minute sliding window, where we're looking at really sort of long-term changes in terms of those relationships. We can also look at it in a 10-second window, where we're looking at moment-to-moment changes. And I'll show you.
But I was just thinking, you could
examples.
extract like different frequencies and sort of find the spectra
Yes.
at each frequency, and then look at the cross spectrum. So then it wouldn't be a question of 10 minutes or 10 seconds. I mean, you could sort of go across the whole spectrum and find out different periodicity.
The tricky part is knowing what it means.
Yeah.
And so EDA is not periodic, unlike heart rate. And so what we'd be finding, hopefully, would be something that said something about an interactional rhythm, which would be really interesting.
Yeah.
The challenge would be to figure out, I think, how we map the findings back to something that will help us understand what they mean. And so I think that would be a really interesting place to do work, and we should talk about it more, because that's not an area that I have a lot of experience, but it sounds like a really...
But when you do this kind of relationship, it's like, suppose a parent is reading to the child, for example. I mean, I don't know whether you've done that.
I have.
And, I mean, you would think that would lead, so if the child is oblivious to, you could surely measure the engagement, I assume, to the physiological activity of the child, right?
You can, and in fact, I will show you an example of that in just a moment. First, I just wanted to show this. Don't worry, you can't read it. I just grabbed this from a thing, just to show the sort of a sort of basic example. So it's a little hard to read on the screen, but essentially, the top trace is showing a student's electrodermal activity over a period of a couple minutes. The bottom traces are showing the I-squared values I was just telling you about. So this is the percentage of interpersonal variance explained for both the teacher and the student. Teachers in green, students in red. And what I want you to see is just the fact that, and basically this is transitioning from a poll group to some independent work, and then there's a teacher who's actually working directly with the student. And what you see is both that there are these changes in the level that you observe. There's also changes in how close together they are and which one is above the other. So you're getting both information about essentially how connected and interdependent are their physiologies during different periods of time and different activities, as well as essentially how different is their level of dependence on one another. And I think both of those things actually turn out to be interesting. But this is just sort of to give you a sort of basic example of that.
Looks like a great feature.
Yes. Well, this is one example that we pulled out because it was a nice, clean example to teach people about this. This is actually part of some ongoing work I'm doing with colleagues at Harvard University Graduate School of Education, as well as with Tools of the Mind, another childhood curriculum.
If the child was non-responsive, you would expect, say, the red line to be kind of flat. Yeah.
I would expect less of that. I think there are exceptions to that rule. For example, I know that with children with autism with whom I've worked, you sometimes can see cases where the child might be responding physiologically, but not behaving the way you would expect. And so they may actually be being influenced by what you're doing, but not show that. And that's part of, I think, what's going on for some children with autism is that it's not necessarily that they aren't reacting to the environment, it's that the way in which they behave in response to it is not the same as the sort of way socially we think of as being appropriate. So the kid may not be looking at you, but they might actually be responding to the things you're saying and the things you're doing. They just don't realize that socially, conventionally, we demonstrate that we're,
Well, could you use these other multiple measuring schemes, like you have the visual
Yes.
technology on top of this technology, the audio technology? There are lots.
Absolutely.
I'm just wondering how, presumably out of that you could form a composite, which would be much more informative.
Absolutely, and I think that's exactly what we want to do as much
But
as
it's
possible.
not done yet. You're saying.
Not using this specific technique. I would say not sort of in the mature way that I think that you're describing it. In almost all of these studies, I'm also collecting video. Unfortunately, I just don't have permission to show most of it publicly especially because we have young kids. People are often pretty sensitive to that, especially if it's kids with developmental disabilities. But I think that there's actually quite a bit of so we are almost always using video, often coded by humans, to interpret these data, and I'll show you, and/or other contextual information. But I think there's a real opportunity to take that the next step and fuse multiple of these sort of computational measures, and that's something I'm hoping to work on the year ahead. So you were asking about sort of a clean example where you control the context as much as possible. So this is what we did for that. A number of years ago. This was done by a colleague of mine, and essentially he collected data from romantic couples. These are college students, and they're brought into a room one at a time. They're asked to sit still and remain quiet for 32 minutes, so there's no talking. It's a very boring experiment for them, during which we record their psychophysiology. In this case, electrodermal activity and heart rate. The results I'm going to show you are for electrodermal activity. For 17 minutes, the first half essentially after a 2-minute baseline, the participants are seated back to back in separate chairs. After they've been sitting back to back for 15 minutes, a tone sounds and they're told beforehand they're supposed to switch, and now they face each other, but they still can't talk. So all we've done here is essentially manipulate whether or not they can see one another. They're the same distance apart, they can't talk to one another, they're in a quiet room where ostensibly there should be relatively minimal environmental variations going on that should be influencing what's going on for them. And what we found was that not only so we found not only did we observe synchrony, so even though they're not talking, just by being in the same space together, we did observe synchrony, but we also found, so that the upper right here is a graph that's showing you the average across all of the dyads that we had of the synchrony at each 2-minute point that we computed. And here for this, I averaged the metrics for each partner to come up with a composite measure for the dyad. And then I averaged that across all of the dyads, and the window there is showing you a confidence interval. So essentially what you're seeing is, so the green box is that baseline period. It's not a ton going on there. Then we have the back-to-back phase, and then we have the face-to-face phase on the far right. And so what you see is that we get a large effect size here in which we are getting much higher on the whole average levels of synchrony when they're facing each other, even though they can't talk and aren't supposed to be otherwise interacting.
What if the face-to-face was happening first, and then the back-to-back was happening
It's a
later?
great question, and if we do the study again, we're definitely going to do that manipulation. This was
Is
done.
both, like heterosexual and homosexual romantically involved companies, or what's the?
I believe these were all heterosexual couples. So this particular study was done by Dr. Richard Palumbo, who's a colleague of mine, and he was doing it during his graduate work a number of years ago. And him and I, every time we get together, are going, oh, we should redo this, and we should also do this and this and this and this other thing. And one of these days we'll get funding and we'll do it. So I think that I believe they were all heterosexual couples. We had a little bit of other information on them, but we didn't collect a lot of that. We really were originally intending this as sort of a pilot to do this, and then it turned out to be a convenient sort of way for us to test some of the different methods that we had developed to quantify synchrony, because it's a nice clean example and because it also replicates a similar study that was done by Ferrer and Helm that's also been published.
Have you done this in stranger couples as well?
Personally, I have not. There are other people who have used different techniques and looked at strangers, and they do find consistently that friends show higher degrees of interpersonal physiology and synchrony than strangers do. And so there are people who have looked at those kinds of things.
Is there any sympathy in strangers at all?
Yes, there is.
And it's not due to any environmental.
So it's impossible to ever fully rule out environmental situations, but I think that I would say my perspective is I think that this is integral to the experience that we may have of, you're sitting next to somebody you don't know on the bus on the way over here, and you strike up a conversation with them, and you feel this synergy or this connection with them, right? In some cases, and in some cases not. And that feeling of that, I think, is actually probably very related to this, and certainly it seems to correlate with self-reported measures of that sort of feeling of connection with someone seems to correlate with the degree to which that's happening. So I think it can happen between strangers, probably because we're wired to do it.
But even if they don't interact,
I don't know if anybody's done this exact study where they didn't have them interact. Most of the friends and strangers literature that I'm thinking of at the moment is, I think they were having conversations. There are some interesting, myself and Dr. Palumbo and some others did a review paper on this a couple years ago, which is one of the papers that's posted online, and that has all of the details about all of the studies and their findings across different modalities and things like that. And I can't bring all of them to mind right now.
Did they ask? You didn't do it in reverse, but I mean, the question is, what happened afterwards? Suppose they actually now separated.
That would be fascinating.
That hasn't been done either.
No. So again, this particular study was done a number of years ago, actually, before I developed this technique, and then wasn't really analyzed or published because they didn't have the technique to analyze this yet. And then when I developed it along with some colleagues, we thought it was the perfect data set to sort of try to address this question of, if you try to control context as much as possible, do you get something that suggests that there's something real here? And so I think what this suggests is that there is, but also that the presence of this is not dependent on interaction. Now, what I would say is that I think that there is a lot to be said for, if we come back to this sort of idea that we're constructing our own conscious experience of affect and of social affect, that in some cases, whether it's caused by a shared environmental condition may actually not always matter. And the example of this I love to use is taken from reality dating shows. It's not really my thing, but I've seen a couple of them, and you have these shows, and it seems like invariably, they take two people, and for their first date, they send them skydiving, or climbing a bridge, or rappelling off of something high. They do this kind of high stress, high arousal thing together, and then afterwards, they just can't stop talking about how connected they feel. Right, and it sort of goes back to the trope about like the two people who get locked in an elevator together in some movie and then they fall in love. Or I'm not suggesting obviously that this means that you fall in love, but I think that there is this piece where to some degree just as with affect, we are making a sort of conscious categorization of the experience that we're having internally, I think something similar is happening with synchrony, and we're making a social construction from that. And I think in some cases, when you happen to incidentally be in a situation where you and some other people experience something similar, say all watching a sporting event together, all going to a concert together, that you may actually interpret that sort of synchrony as a feeling of connection to that person, even if there may not actually be anything about you or that person and your sort of shared experience that's changed, really.
Do you think it's a measure of quality of a relationship? I mean, could you use it to predict split up?
You can, and that was done back in the, I think, late 70s by, I believe, Gottman, who used a different technique for measurement, but the same idea, and looked at the physiology in married couples during arguments, and actually found, interestingly, that couples that had a higher degree of synchrony actually split up more. And I think the important thing, that I think highlights the importance of the context. So the degree of synchrony that you have during an argument may actually speak to whether you're driving each other's arousal level up, right?
But it could be bad for the relationship altogether, that we don't need.
Right, and there's the concept of stress buffering in relationships, right, where one person doesn't react, and that actually can have a positive co-regulatory effect on the dyad. So I think that, so to answer your question, I would say, I think there is definitely some really useful and interesting information there, but I wouldn't say that it in and of itself provides a relationship quality metric. I think it provides a useful descriptor that one could use with some other knowledge to maybe try to assess relationship quality.
Elicit the other knowledge with some of the other techniques you were talking about earlier.
You can elicit some of it. I mean, the big thing that they find is
sound. I mean, for the argument, for example, you could probably get a sense of tone of voice, right?
yes, and Gottman, I believe, used to claim that he could overhear people, or watch people at a diner having an argument and tell you whether they were going to get divorced. At least that's the story. I think that I know that one of the big findings there on the nonverbal side is contempt, and facial expressions associated with contempt are one of the things that seem to be pretty high predictors of relationships that don't work out. So I definitely think that bringing this together with some of those other pieces could give you a lot of information there. And then just very briefly, I wanted to show you two or three study examples that I'm going to go through kind of quickly here of ways that I've applied this method and these data that I think are applicable to this group. This is a study that I did last year, funded by the Simon Center at MIT. And this was essentially comparing interpersonal physiological measures, the stuff we've been talking about, between a child, a parent, and a researcher with existing behavior and informant report measures of social reciprocity in young kids. These kids were split between ASD and typical, but I'm not going to focus too much on the differences we found there. But we found interesting differences sort of throughout. So basically in this study, we were measuring physiology. We had a bunch of cameras to capture context. And we did a series of different things. So we had them read a book with their parent. We had them play with age-appropriate toys with their parent. We also had them interact with a researcher who was doing a sort of standardized test that probes for social reciprocity that's used in diagnosing autism a lot. And we were trying to use that as sort of a standard probe. So even though obviously you're interacting a little differently with each kid, the researcher's following sort of a set script with set toys and a sort of set interaction that they're carrying out that's been previously validated. So we looked at two things. One was we looked at the parent in a bunch of different places, then we looked at the researcher and the child during this sort of period where the researcher and child were interacting for 10 minutes. And we found two interesting things particularly there. One of them is that we found a pretty high correlation with this social awareness score from this standard measure called the SRS-2, which is a social reciprocity. So it's an informant report measure. It's being filled out by the parent, and it's about 40 questions long, and it has a couple of subscales. This graph on the left, which again I apologize, is a little hard to read here, essentially what it shows you is that the level of interdependence between the child and the researcher was inversely proportional to their social awareness score, which is what you'd expect because the social awareness score is an impairment score. So values more to the right these children have more impaired social reciprocity for their age. Values more to the left, they have less impaired social reciprocity, and so you can see that you get a pretty strong correlation there. One of the things that I think is worth noting here is that this is actually all typically developing children. So this was using a measure that we were originally going down this route because we were interested in looking both within but also across typical developing kids and kids with ASD. But these are all kids who scored within the typical range. So even within the typical range, we're actually seeing that we're quite sensitive to differences in sort of their social reciprocity. And again, this is a global measure, so it's not asking parents to rate their child's social reciprocity during this 1 hour that they were here in a room interacting with us. It's actually asking about their ability generally. So I was pleasantly surprised to see the degree of correlation that we got here. And then we also had good findings, albeit with a slightly smaller sample size, in the ASD group, where we were using a measure that was particularly looking at sort of empathizing ability. And again, here we found a pretty strong correlation between children's score on this informant report measure that's being filled out by the parent as to what their empathizing ability is and the degree that we're seeing during this sort of brief play interaction with a stranger. That was interesting. And then I want to show you another study that I did a couple of years ago. This is a study done with colleagues out at UCLA, with Connie Kasari and her group out there. They work with children with autism who are minimally verbal, and they have a therapy that they've developed called Jasper that's all based around sort of building children's capacity for joint engagement, which is something that's tied to later language development. So we went out, we collected data this is really pilot data from 6 children with an autism diagnosis and their therapists during a sort of 45-minute long session here. Aside from putting sensors on the therapist and the child, we weren't doing anything else to change their interaction. They basically carried out what they would normally do, and then they did behavioral coding blind to any of the physiology data using an existing coding scheme that they and colleagues have developed that looks at engagement state. And again, I don't want to go into a ton of detail here, but we can talk about it afterwards if people are interested. But essentially, we compared 2 different levels of behavioral engagement that were coded. So these are coded second by second throughout the interaction. And then we binned the instances where the therapist and the child were engaged in coordinated joint attention engagement, which is a higher level of engagement in their model, or supported joint engagement, which is sort of a lower level of engagement, and then unengaged. And then we compared the I-squared values that we were getting during those times. And what we found is that, as you'd expect, the coordinated joint engagement was associated with higher levels of interpersonal interdependence, so I show this primarily to point out that in addition to sort of global things like the measure I was showing you with the N-Train study, you can also use this to look moment by moment to try to look at how this is changing over time, which is one of the things that most of the existing literature that's looked at this doesn't really address, but our findings really suggest that this is something that's very fluid and changes over time, and that those patterns may be really meaningful. And then finally, I just wanted to end by showing, so one of the challenges as we go from dyads, as I've been showing you, to a large group, is that you now get a multidimensional matrix that has a lot of information, and you need to be able to really take that information and try to start making sense of it. And so one of the other things that I've been working on is some ways to visualize and quantify this. So this is just one thing I wanted to show, but essentially this is a visualization that I'm showing here with 4 individuals, a teacher, and 3 students. And this is capturing a couple of different things. So the sort of connections between them here show something about how they're influencing one another. The thickness of the outside arc shows something about how that individual's arousal is changing over the course of the day or the interaction. And then the width of those individual chords, those little strains that connect the arcs, that tells you about the magnitude of the influence. So how much of the people in this, how much is one person influencing the teacher versus the others? And then the gradients that you see here tell you about the direction. It's a little hard to see here, but there's actually little particles that are flowing here. So you can see, for example, that number 68 here, the person in blue, is influencing both the other two people, the other two students, more than they are influencing that student, but is being influenced by the teacher. And in fact, the teacher appears to be sort of influencing all of them more than he or she is being influenced by the students. And this is
How do we get this directional causality? I'm sorry. So before you were looking at these cross correlations. Now you're looking at, so how,
It's comparing the differences between, so remember how we get an I-squared model, we get this I-squared figure for each dyad, so for me predicting you and for you predicting me. And those values are often correlated, but not identical. And so this is comparing them.
so it's a relative strength of the
It's a relative strength thing. And this is not meant to be a confirmatory way of portraying any of this. It's meant to be something that's illustrative for sort of looking at this data and trying to think about qualitatively what's going on to try to generate hypotheses about the type of dynamics that are occurring and how this may relate to the interactions we're observing. And so this, for example, is an example of this every 2 minutes, I think over a couple hours for a classroom. And so you can just see how complex this is, but also how much variability there is in terms of how people are influencing one another. And I think that, so I hope this speaks to the richness that I think this type of data can provide in making sense of what's going on here. And obviously there's a lot of work that goes from being able to measure it to being able to interpret what you've measured, and I think that's one of the places where getting more data for us is going to make a huge difference, because I think the more we
So
can collect
if there's
this data,
no flow in this regard, you want to think about using this to, when you get to many students in the classroom, to form peer groups maybe, or some sense, or groups that the teacher is referring to more than, could be their problem kids or their...
yeah, so I think one thing you could see from a diagram like this is which children are having an influence on the teacher,
Right, and the teacher.
and which children are being very influenced by the teacher. And then you can also see which children are influencing each other. So I can tell you, having done this in classrooms with really young kids, you can see instances where one kid starts to get upset or for whatever reason, and that would ripple through to other children in the class. And so you would be able to see that using this type of a visualization. This is just one of many ways that these data can be represented, but I wanted to show it just because I think that it really illustrates how much is going on and how complex these interactions are, but also how rich they are. And one of the things that continues to surprise me in these data is how significant it appears other people's affect are in determining the affect of those they're interacted with in a context, much more so than I expected when I first did this. And obviously you can't control for all of the shared environmental influences and all of those things, but I think that you do start to really see that this is a very significant, that the social component is very significant.
But so far, these are contemporaneous relationships, if I understand correctly, right? I mean,
yes.
And so I'm thinking in terms of like a dynamic, more dynamic system, you could imagine almost like an impulse response function. Teacher affects this student, student affects another student, student goes to another student, and then maybe back to the teacher. I'm just wondering if you could trace that out. Has anybody done that?
So I have not done that computationally. I don't know that anyone has taken this specific interpersonal approach. The closest thing I can think of off the top of my head is there was work that was done by Mahler, I think, in Europe that looked at choral singers. And so they recorded physiology from choral singers and from the conductor. And they actually did find this sort of directional flow from the conductor to different choral singers and then out throughout the chorus, like during singing and these sort of things. So I think that, I definitely think that that is there. And I think that one of the interesting challenges here is to try to take these data and then do modeling based on that and based on what we know from other sources of information and from theory, to then use this kind of data to test out models for what that function may be and how that may relate to other things of interest, like the classroom climate, for example.
This is, I think it's really interesting. It looks like the mechanics of social network and how we talk about contagion. There's a big study in Framingham. A sociologist said, like, if your friend's friend is overweight, your chance of being overweight is like up by 10%. And this kind of shows, to me at least, this is how like we influence one another.
Yes, no, I think that this is very much the way I look at it is as a substrate of sort of this.
But it's not quite how. I mean, you're not going to talk about causality here. You're not going to. Until we sort of put some dynamics in here, I think it's going to be hard, right?
I agree. I would not suggest. I think it's yes. So I wouldn't say it doesn't tell us necessarily the mechanism for this, but I think that it is measuring a phenomenon that is related to our perception of this social experience. So I think that these physiological changes are related to the way that we're perceiving ourselves and perceiving ourselves interpersonally, and that they have a very big influence on that. And so in that sense, they're sort of a substrate. But I agree. I mean, I have my own theories about what the mechanism underlying the things we observe may be, but I think that it's too early to be able to say with confidence, and certainly not from these data alone, what that is.